Here hex 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 hex. Here hex 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 hex. Here hex 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 hex. Here hex 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 hex.
Hey everybody, welcome to another episode of the Guild. Today we have a wonderful guest from Central Texas. Everybody show her a warm welcome and please place your comments for the subject matter. It's soil web. And so this is Laurie. Welcome, Laurie.
Thank you. Thanks for inviting me. This is going to be fun. So I'm going to talk about the soil food web. I think you called it just the soil web, but it's the soil food web. And it's the slides I'm using are actually from a microscopy workshop. So there's a few concepts that we're going to cover first. The first one.
What is microscopy?
So microscopy is just using a microscope to look at the soil organisms. Technology has developed with microscopes, so we can use, we're actually putting that soil sample under the microscope and looking at those microorganisms live. And we can do some pretty cool stuff with that. So let's first talk about what we're going to be looking at. We have let's see, do I need to share my screen? Oh, you got it. Okay. So first we're going to talk about this cool word, holobiont. And you can just see with every kind of organism, it's actually a community of organisms. So you can see on this slide, it's comparing different microbiomes. So we have the air microbiome, the phylosphere, which is the leaf surface, endophytic, which is inside the plant. There's a biome inside the plant. Rhizosphere, rhizos is root. So you have an assemblage of organisms in that rhizosphere, and that's the rhizosphere microbiome. And then, of course, we have the soil microbiome, and that's mostly what we'll be talking about today, but we are going to zoom in and be talking about that rhizosphere also. What do you think? Got questions on that?
Not as of right now.
Okay.
That's a great illustration.
Thanks.
The little areas of life.
Yeah. So we'll go on. I'm not really going to talk a lot about this. I'm going to assume with y'all's background, you know the issues that we're facing today, why our soils are degraded. You can see the tilling, herbicides, pesticides, blah, blah, blah. We're just going to blow through that. So here's the soil food web. This is what we're going to be talking about. And the soil food web is just all about any kind of food web. It's predator-prey relationships. And they call it a web because when you're talking about those different trophic levels, first trophic level is going to be the, lower life organisms. They get eaten by the next trophic level. They get eaten by the next trophic level. It forms a kind of web because there's a lot of, crossing over. I mean, some organisms eat multiple organisms and so forth. So this is central to understanding the soil food web, this diagram. I think maybe here I should give y'all a little background about the Soil Food Web School and Dr. Elaine Ingham. She's the one who kind of really spearheaded the conversation about the soil food web. This diagram right here was published in a USDA publication back, I believe, in the 80s. And then she eventually kind of got ousted by academia and formed her own school. And that's where I'm certified through the Soil Food Web School. She will certify to do the microscopy work. And that means that we're doing biological soil assessments rather than chemical, and we'll talk about that more later. So again, this is just kind of talking about the degradation of our soils. And this is kind of an important concept here, is just that there's succession. Succession just means that plants, you have lower complexity plants that are going to grow at initially, and then they go on up in succession to, a rainforest. And so lower succession plants are going to want what is called a bacterial dominant soil. And you can see these ratios down here. That just means in the soil microbiome, there's lots of bacteria. And as you go up, that fungal to bacterial biomass is becoming more fungal. And so vegetable crops usually the basic ones want a 1-to-1 bacterial to fungal biomass. And as you go up to the rainforest, you can see that that fungal biomass is really high. So that's that. And what
went through my head was pioneer plants.
You got it.
Over
exactly
there with the
right. Pioneer plants, you could even say weeds. They're called ruderal species. Whenever you have a disturbance, that's what's going to come in first. And it's because there's been damage to the soil microbiome, and you're starting back at the beginning with lots of bacteria. And then you go up in complexity. Yeah. So this is important. I don't know. I mean, I speak to growers. I also speak to gardeners, and then my background is landscape design, ecological landscape design. So we use a lot of native plants. So we're sticking down here with native plants, but probably even here, but with growers that are growing food crops, they're going to want at least a 1-to-1. You go up. Most food crops want the higher ratio. And then, of course, if you're growing shrubs or trees, fruit crops, tree crops, then you're going to want more fungal dominance. So now we're going to kind of, I think it's important. We're zooming into the soil. So we're looking at really, really tiny things. So now we're going to kind of zoom out, and we're going to look at the carbon cycle. You guys probably all know the carbon cycle.
With that last slide though, where is a whenever people say food forest, is that an oxymoron?
So yes and no. I mean, this is very general, because we are talking about an ecosystem, and an ecosystem is dynamic. And as we zoom into these to these organisms, they change their function. So in a food forest, you're going to have all kinds of what would you call them? Like microbiomes, little small, like when you create microclimates in a growing space. Are you familiar with microclimates?
Yeah, we are some here in this group.
Yeah. So basically you have a space where you're growing, and within that space, depending on whether you build structures that create shade, you have more sun, you have wind blocks, then you're creating those microclimates, and that soil microbiome is going to adjust itself.
So it's almost like this diagram that we're looking at is kind of just only a linear thing, when it could be just sort of a wavy or S thing. Yeah.
Yeah.
Cool.
I think, yeah. So with the carbon cycle, you have the different pools, right? You have carbon stores. I mean, carbon is cycling in the system. You have the oceanic pull, you have the atmospheric pull. We're talking about this pull right here, the soil carbon pull. You can see there's even a plant biomass pull, but that's above ground. We're going to go below and we're going to talk about the soil carbon pull. How does that carbon in the atmosphere get into the soil? Hopefully we all know this. It's photosynthesis. We've all seen this picture, but the plants have the capacity to take the energy from the sun, the CO2 from the atmosphere, which, by the way, are the only things they cannot get from the soil. So they pull them from up here. It goes through the photosynthetic process, and then it has exudates, it's called, which are the carbohydrates that it makes, and proteins also. And those go into the soil. And that's photosynthesis, taking that carbon up here and storing it down in the soil. So there's a phrase coined by Dr. Christine White. I think I got her name wrong. But anyway, she calls it the liquid carbon pathway. And so let's go back to that picture and think about a liquid carbon pathway. It's the same thing, right? But think about that carbon going all the way down into the soil. And see, the microbes are feeding off those exudates. The microbes are collecting nutrients in the soil, nutrients that, you know, in chemical ag, they'll say, yeah, you've got phosphorus in the soil, you've got calcium, but it's locked up. The plants can't get it. Well, microbes get it, and they trade those nutrients for the exudates that the plant is pumping out. And that's how the plant gets, continues the cycle. I always like to say, you know, you can look at the sun all day, but you can't make hamburgers by looking at the sun, right? But plants do that for us. They make food. They take that energy from the sun. They take CO2 in the atmosphere. They break apart. They break those bonds, and that carbon gets up here, down here. So now, so we just zoomed in. We zoomed in. We were looking at the carbon. I don't know. Can y'all see my paper here? It's kind of silly. It's just 3 concentric circles. The carbon cycle is the outer circle. Photosynthesis, we're zooming in. Now we're zooming in even further. Now we're looking at the roots. This is the rhizophagy cycle. This is a root tip. Well, this is a root. This is the tip. It's called the meristem. Around this tip is the rhizosphere. It's all the bacteria and fungi that are right in this place around the root tip. The plant is sending out exudates, and it's changing those exudates based on what it needs. So it's basically saying, hey guys, I need this. You want, I don't know, Jeff Lowenfels, and we'll talk about him. He says, you want Japanese food? Here, I got Japanese food for you. It attracts the microbes that it wants to attract. And they go through the meristem tip, and they go into this periplasmic space here. This is a very thin wall. That's how they're able to enter in through there. The plant, there's some good stuff goes on in there. The plant breaks down the cell walls, releases those nutrients, and then these are root hairs. And we've been taught for centuries that, these root hairs are extending the capacity of the root to get nutrients from the soil. That's not what's happening. Those root hairs are growing as pathways, exit pathways for the bacteria. So their cell wall has been broken down. these root hairs grow based on the bacteria that are exiting. The bacteria are sent back into the soil. I should be using their right here because you can see the cycle. They're going back into the soil, rebuild their cell walls, get more nutrients, and there you go. And that's the rhizophagy cycle. Right here, I love this picture right here. If y'all have never watched Ray Archuleta, you should look him up. He calls this liquid sun. So this is a real picture of this diagram. Those are the exudates going out. There's bacteria all around there. He calls that liquid sun. I love this picture. This is from Dr. James White. That's in this book, Teeming with Bacteria. Oh no, can y'all see it?
So, yeah.
Yeah, go ahead.
Wait, can you hold up the book again and
Sure.
give us a good view of it with the camera? Like the camera does adjust. Yes, that's great.
Okay.
So it's really interesting to me how there isn't a fine line between things. Instead, it's a membrane after a membrane, and then there's a society. And then the oozing out, as you're saying, the excretion.
Mm-hmm.
That's tremendous. And then there's forward thinking there on the tip. There's forward thinking. The plant says, I'm going to need about this amount here soon. And then maybe I didn't quite catch how it changes what it is that shows up at the tip, but it's thinking for itself or planning for the future.
Okay. So you didn't catch it because I didn't say it, because actually we don't know. That's yeah, there's scientists all over the world. We don't know how the plant is communicating that. We know that the plant is doing that because you can see this process, but we still don't know how. There are theories, but we don't know how. This picture is so beautiful to me because this is the root tip. This is the rhizosphere. I don't know even what kind of microscopy this is. I think it's a kind of laser microscopy, but to me, that's like light right there. And these are the exudates kind of leaking out of the root. And if you think back on the carbon cycle and photosynthesis and the fact that this is liquid sun, it's like light is going into the soil in a place that seemingly never sees light. But the microbes see light. And I think right here is a good place to talk about
So
go ahead.
That's darkness there.
This is the soil. Yeah. This is a root. This is a root tip in soil. I mean, this is a microscopy. I mean, a slide. So no, the soil's not here, but yeah, this is what happens in the soil. That's the root tip. That's the meristem. This is the rhizosphere. That's what happens in the soil.
Okay. And you said it was a good time for us to mention
Epifluorescence. So with microscopy, the kind of microscopy that we do is the technique we use is called shadowing. And shadowing just allows you to kind of allow these microorganisms to pop. It's just using the microscope in a certain way that you can see that bacteria. But there's also a module that you can put on the microscope called an epifluorescent module. It allows you to see the cyan blue wavelength. And these bacteria and also the arbuscular mycorrhizae that form relationships within the root, they autofluoresce in that light because of the phosphorus. So they actually glow. And that enables us to see not only the stuff happening in the soil, the microorganisms in the soil, but we can begin to see those symbiotic relationships that the fungi is forming with the root. So this is where I kind of want to hear from y'all, Davin, to see where you are. What kind of questions do you have now?
Well, one question that popped up in the chat was, what do you recommend for raised beds?
So I probably need a little bit more information. What do you mean? What do you recommend? Like what kind of soil? Like how to build the raised bed? What more specifically? What is
Recommendation for soil?
Yeah. So honestly, I don't recommend raised beds.
Okay.
People use them a lot because they feel like their soil is poor. Their soil is poor. Their soil is degraded. What I recommend is you cultivate this soil food web in your soil so that you have healthy soil. Well, what's the other?
And the guy who asked that, he's typically not in our group. I was just chatting with him on Twitter and said, hey, this is going on. So what I've typically seen is that people completely just buy brand new soil that see this soil that comes in a bag. It has a certain lifespan to it because it was picked up from a place, put in a bag, and it was calculated. So it may last for only 3 years for what it is that you're interested in doing. And I also recommend that folks, if you are creating a raised bed, which is an artificial environment that we're not really talking about here right now, make it to where it's right outside your kitchen door, because that makes it the most easiest to maintain. And and the guy who asked that question, he's a chef. So
perfect.
Yeah.
He needs an herb spiral. So I will say a little bit more about that, because what do we do with this? What do I do as a microscopist with this information? So we do right now several projects that I'm involved in is actually assessing off-the-shelf products. So we're not so much looking at the soil products because we know those are crap. But we have been looking at a lot of compost. And so we can do side-by-side comparisons, and all compost is not created equal. And most of the compost that you're going to buy, in any retail place is going to be bacterial dominant. It's not going to have a lot of life in it. So I don't know. I mean, should I run through these soil health principles?
Yeah, sure. Because
you're all you have growers. Mm-hmm.
Yeah, go for it.
Okay.
Do your presentation.
Yeah, you can stop me whenever. So the soil health principles are common. They're all over the internet, and they'll have anywhere from 4 to 6 principles. And we're just talking about soil health as the capacity for soil to function as a living system, right? It has these symbiotic relationships with the plants. Generally the principles are cover the soil. It's called oftentimes soil armor, continuous roots in the ground, increase biodiversity, minimize disturbance, integrate livestock. I love that some add this, know your context. And actually that should be number one.
What's that mean?
Know your context. So for example, a question about raised beds. I could throw out general recommendations all day, but I don't know your context. I don't know what you're building on. I don't know what your resources are to build that raised bed. What are you going to think about lining it because you have weeds everywhere? Do you have animals that are getting into it and defecating in it? You need to know your context.
One of the permaculture principles is to live on a property for a year so that you are able to examine enough of it.
That's right. Yeah. So I'm not going to talk about each one of these in detail. Y'all can look at them, and if you have a question, I'll definitely address it. But the two that I really want to emphasize is the first one, cover, cover, cover. And, I speak to growers and I like to always ask them, here's a knowledge check. What am I referring to? Well, it's that first principle, which is cover the soil. They oftentimes call that soil armor. Next question is, okay, a reality check. Who's covering their soil? Many people know these soil principles, but are you actually doing them? Are you actually covering the soil? Why is cover important? Well, think about the elements: sun, rain, wind. So covering the soil, I always like to bring it back to the microbial community, that soil food web. Those are your babies. These are growing phenomenal crops for you. There's all kinds of research, nutrient dense crops, but you have to protect those babies. If you have sun beating down on bare ground, what's happening to the microbes? You're frying them. You have rain beating down. What's happening to those microbes? Well, one, the soil is getting compacted. But what does that mean? That means that pore space in the soil is getting smashed, or it's getting filled with rain. You're basically suffocating them. There's no oxygen in the soil. Wind is pretty obvious. Bye-bye, babies. They're blowing. The wind is just blowing them away. The next one, let me say something about this picture. This is interesting because Ray Archuleta talks about he's like, everyone thinks about covering the soil and they think about cover crops, right? And he really doesn't like the name cover crops because they're actually doing so many other things besides covering the soil. And we call those ecosystem services. This is a book from 1911, the entire field completely occupied by crops, rendering effective service. These are principles that they knew. They knew them really before the Industrial Revolution, 1911. There's a lot of good literature from the mid-1800s to before, the wars, when they started pumping out the chemical fertilizers. So the next one that I really like to emphasize is roots in the ground. The principle is continuous roots in the ground, 24/7, 365 days a year. This is kind of an exaggeration because we're in different climates. Not everyone can have roots in the ground 365 days a year. I think we can where we are, Davin. But, the point is to always have the roots in the ground. Why? Because you are not capitalizing on that photosynthetic potential that those roots have. Think about it. Through photosynthesis, you saw the roots. That's how it happens. You cannot have healthy soil without healthy plants, and vice versa. That's the holobiont idea, is that this is a community that all works together to function on behalf of the whole, and you can't separate them. So you need healthy soil for healthy plants, and you also need healthy plants for healthy soil. So this is some more, a statement by Dr. Christine Jones. She's the one that coined the term, the liquid carbon pathway. And she makes this point that the plant root inputs build soil carbon 5 to 30 times faster than carbon derived from above ground biomass. So this is just stressing that point. You need roots in the ground. You know, a lot of people that know about covering the soil and soil health principles have above ground biomass, right? They're growing cover crops, they're doing chop and drop. But if you really want to speed up that process, have roots in the ground. They're made. You saw the rhizophagy cycle. So this is where I'm gonna stop, and then we can have questions. Oh, actually, I'm not gonna, anyway, let me cover this slide. So when you're thinking about soil health, these are all the things. So these would be the services that the soil food web provides. And what I like to ask people is to look at all these things. Where are your hangups in your soil? You know, do you have a problem with water infiltration? Do you have puddling? Do you have runoff? I mean, those are huge problems. Well, that's a problem with your biology. Do you have a problem with compaction? That means you have no aggregate formation. So that's a big problem, and it's a problem with biology. So all the soil food web is completely focused on what we call a biological approach versus a chemical approach, right? Because all the nutrients, except for the CO2 and the energy that your plants need, are in the parent material of the soil. I mean, there's going to be a few, strange soils that maybe have no boron, no molybdenum or whatever, but those are rare, and then you go in later and you fix that. But for the most part, all soils, wherever you are in the world, have everything that they need nutrient-wise there, but it gets locked up. And through that rhizophagy cycle, you have that nutrient cycling. So these are all the things. And I think here, if anyone has questions, we can stop, and then we can start looking at the microorganisms.
So I'm going to read through chat. Welcome, Finbear.
Hey.
Finbear, this is Laurie.
Hey,
Hi.
Laurie. Nice to meet you.
Yeah.
It's been pretty interesting. I've been listening in whilst fighting with my new chair that just broke to its pieces as I was getting ready, so I had to put it together the second time, and hopefully for keeps this time. I was watching Paul Stammet giving a lecture on, I think he was talking about how I don't remember, I don't think it was any of the cordyceps that he likes to collect, but he was maybe growing something like lion's mane or something. And he said that after he had all the flushes out from it was just like this mushroom growing kit that he had.
Right.
I guess they sell them from the website. And after the third flush of mushrooms, it was clearly running out of energy, and he just dunked it in a bucket that had like waste oil from his car.
Mm-hmm.
And it took the mushroom or the fungi a couple of days to find out what kind of like polyaromatic hydrocarbons are present. And once they sort of cracked the code on how to break the chains, all of a sudden the entire grow box started producing another flush of mushrooms. Of course, not edible anymore because of all the heavy metals and stuff in the oil, but still it found a new source of energy. And that's been something that's been really interesting that I've been sort of pondering, is that would it be possible to use mycorrhiza for, like, say there's a an oil spill somewhere, or somebody has a gas station or something, and there's a leak in the tank for like 10 years, and it leaks into the ground under the parking lot of the gas station. It's gonna be a terribly expensive deal
Clean
over
up.
here in
Mhm.
particular. Like, you have to dig a huge hole and change all of the dirt for like tens of meters, which is like, it's quickly like 30 to 50 feet deep that you have to go and like change
Yeah.
the soil. I was wondering, do you think it would be possible to use fungi to remediate the soil, like first inoculating like a deep bed of mulch, like wood chips or something, and then just covering the entire area for something like a year or two with the
Yeah,
mulch?
definitely. 100% definitely. And people are already doing that. Fun, Fungi are known to be great shapeshifters, and mycologists that I've listened to, they often talk about training the mushrooms.
Yeah.
So, and in the area, so there's a lot of research being done on that. There's already products on the market that are kind of zoning in on specific remediation type needs. And in our area, we have we have a very active mycological society. And so they've partnered with the mushroom growers. Mushrooms are they're very popular right now. They have a waste stream, which is what you're talking about. It's basically those blocks. They're called SMS, spent mushroom substrate, or just spent mushroom blocks, and you can collect them from mushroom growers. They're often very happy to offload them because it's a waste stream. And some mushroom growers might try to compost them, but it's a huge undertaking because it's a big waste stream. So I have been collecting. I can get, I don't know, 50, 75 blocks at a time. And I have been doing experiments and looking at that microbe community and how it's changing. And mushroom blocks are really good to, even if you break them apart, if you have access to them, you can do what we call low tech gardening.
Yeah.
You're breaking them apart. You're maybe giving them a food source, which is carbon. So cardboard is good. Coffee grounds is good. And crumble them apart, mix them with, leaves, whatever, and you will get very nice garden soil. Again, in time frame, it depends on your environment. In our environment, we have a lot of heat. We also get drenching rains sometimes. So carbon breaks down very quickly in our environment. So I did a bed in July and tracked the microbial community, and I just did a sample a few weeks ago, and there was so much activity. You have all the trophic levels, and we'll get into that after this, but basically you have the bacteria, you have the fungi, you have the protozoa, you have the microarthropods, you have the macroarthropods, and you have plants that are just happy as can be. I don't really water. I have an irrigation system, but I turn it off. I had to water in the drought a few times, but now this summer I'm turning it off. If the plants can't live without water, bye. So
Yeah.
yeah, that's a great question.
Yeah, that's been something that I think we've talked about this with Eco Builder, like way before, back during the early streams. I might have already mentioned it, but it's something that's been on my mind for years. we got plenty of old gas stations over here and all sorts of areas that because like you could buy a lot of land for almost nothing, and that's really close to city centers nowadays. So
They're in
actually,
Finland.
yeah. So a bit far away from you guys, but that's a problem with you Americans living so far away.
So Hazel Hex is in the chat saying that they cover their soil with straw. It keeps in the humidity during the summer. They
Yeah.
don't rototill, and he wonders what you think about planting broad beans like fava in the soil to bring nutrients deep down up to the surface.
Yeah, definitely. I mean, that's a common practice. It's a proven practice. Just to get those roots in the ground. The fava beans are going to have the rhizobacteria that they form the nitrogen modules. So that's a popular cover crop to use. But really, any plants. I mean, you could use cereal crops. You could use brassica crops, and eventually you would want to go up though, because they don't form those mycorrhizal relationships, not as much. But one thing I wanted to say about that though too with the straw, and I don't know where the question is coming from, where they are, but in all over the US, straw has become an issue. Well, straw and hay, more the hay, but it's contaminated. And because there's not, I mean, maybe the source, they're not applying herbicides or pesticides, but there's so much herbicide drift that things are still getting contaminated. So the feedstocks that we're using to do different kinds of composting are becoming harder to come by to get really good feedstocks. And so a lot of times we just encourage people to just grow your own.
Yeah, I
He
think I
says
heard
he's in
about
France.
In France, yeah, I would imagine they're probably having the same issues there.
they
with
don't
contamination.
no the French are really strict with their
Are they?
yeah, they are. They're very particular about what is allowed to be sprayed. So, like the French are the only country in Europe, I believe, that doesn't allow Roundup at all.
Wow.
And so that,
Hazel, please correct me if I'm wrong, but I think well, if I'm wrong, at least I'm not lying.
and that brings me to one of the things that I highlighted in my brain from our last conversation, Laurie,
Yeah.
is how you recognize you're seeing firsthand the recycling issues that we have here in Central Texas, and how plastics have just been mixed and matched and put everywhere. And so
Right.
we're, where are the right sources? What should we be looking out for?
Oh gosh, that's when you come to the matter of plastics, there's almost no escape.
Didn't you tell me about some lady who had to like remove all of her topsoil? Or maybe that was a Paul Wheaton story.
It may have been, I mean, it's happened for sure, because the plastics, I never can remember the name. I always think PCBs is the acronym, but I don't think that's the same thing. But it's basically what they nicknamed the forever chemicals. And they're just very resistant to break down, and they abrade to very, very small particles. So they are everywhere. They're in the rivers, at least in the US. They're in the rivers. They're everywhere in the environment. They're in our bodies. they're probably doing research everywhere, but I'm only familiar with what they're doing at Texas A&M, where they're making headway. They're finding fungi that they're basically training them, and they're getting close to finding something that will process those forever chemicals more quickly. But I also wanted to mention, like, there's so many products on the market right now. I was trying to find that in our area, we have a company, and I was just talking to their distributor yesterday. They have a bacteria that they have in one of their products, a specific strain of bacteria, and the research is documented. He showed me the research. It breaks down glyphosate. And that's an on-the-shelf product where we are. So that kind of stuff is exploding right now because of all the new information about the microbial community. So there are products everywhere. But the thing is, I mean, we kind of nickname them bugs in a jug, right? A lot of times you're not sure what they're doing because people don't know how to track. You're applying, it's basically a land management decision, right? You're applying some kind of amendment or biostimulant or mycorrhizal spores. But how do you know if you're getting the biological effect that you want? Well, that's where the microscopy comes in. And let me see if I can. Yeah. So these are unless y'all have more questions, you want to go on to the microbes?
We just got some feedback from Hazel over in France. He's saying that they don't have the cut green lawns that we're used to here in the nuclear family states. So everything's like wild turf, grasses, dandelions that you can eat straight out of your lawn.
Another
Awesome.
reason to move to France. I have to fight wars with HOAs just to get rid of lawn and plant a wildflower meadow. I mean, yeah.
I can't believe how much say a neighbor has over there on what you can do on your yard. It's amazing.
We're starting to see these videos where people, they have their door ring cam or whatever, and so after an hour of a package being on somebody's front porch, their neighbor comes up and says, this is an ugly package. You've had it out here for an hour. I'm gonna report you. We already told you the rules. We're kind of getting tired of this.
Yeah,
that's too much, man.
So here are the key soil microbes. Of course, the first thing we're looking at is the bacteria. And you can see, let me get my cursor. These are when we're doing the microscopy, we can't identify to species. You'd have to have an electron microscope. You'd have to be in a university or wealthy enough to buy these, I don't know, they're $20,000 up to the microscope that I have at A&M is $1 million. But with direct microscopy, we identify by morphology, so their shapes. So you can see the coccus when you're looking under the microscope. It's just a little circle. You can see rods, which is bacillus. You can see, shapes like this. So when we're looking at bacteria, some bacteria are aerobic. They live in an aerobic environment. They need oxygen. You have to have at least 6 parts per million oxygen for them to survive. And then some bacteria are facultative. They can kind of shift. And among the facultative, you have some that can only shift aerobic, and some can only shift anaerobic. And then you have bacteria that indicate an anaerobic condition. If more oxygen comes into the environment, they die. So that's bacteria. We look at actinobacteria. which are strands. Basically, they used to call them actinomycetes because they look like fungi. They're long strands, but they reclassified them into their own category. They're actinobacteria. Actinobacteria are everywhere. They're the little, if you have a compost pile and you see a kind of dusty white material, not the white mycelial strands, but a more dusty, and you pick it up and it kind of breaks apart, that's probably actinobacteria. If it's white and it doesn't break apart and there's kind of strands that hang on to that organic material, that's fungi. So again, I mean, those organisms, when we see them, they're going to give us clues to what's happening in your soil. And what we want with the soil food web is we want an aerobic community. In our compost piles, we want an aerobic compost pile. These are some of the guys that are going to indicate anaerobic conditions when we find them. Again, and this is just going to reinforce that point more. What are some of the characteristics of a bacterial dominant soil? You're going to see poor structure, no aggregation, it's compacting easily. And what that means is that you don't have those passageways through your soil. And the passageways, not only do they allow gas exchange, which is hugely important, but it's also where the roots grow. It's where the microorganisms use them as pathways. So you need structure to have healthy soil. It's going to grow the lower successional plants. So you don't want to say it's bad. Maybe you are growing lower successional plants. That's what you need. I grow natives. I have to tell installers all the time, no, do not improve the soil. Because if you improve the soil, the native plants won't take and the weeds will come in because they like that, a little better. And then it produces nitrate. Again, that's going to even cause it to go more anaerobic.
You're getting that funky smell from anaerobic.
Yes. Yeah. The anisophoric. Yeah.
Yeah.
Mm-hmm.
Or like rotten.
Yes. Like rotten eggs. Mm-hmm.
Yeah.
So the next group, that's bacteria. Now we're going up to fungi. Fungi are going to feed on the bacteria. They're also going to feed on the more complex foods. So the exudates are actually carbohydrates and proteins and amino acids. Fungi breaks down the more complex foods. And so you're moving up, right, in succession. Let's see. They're easily destroyed by tillage and, of course, chemical applications. We also look at the oomycetes. They're also a kind of fungi, but a lot of the pathogenic fungi are going to fall in this category. So just remember those microbes. It does confuse some people when they think about what? These microbes are going into the root. Aren't they pathogenic? No, they're symbiotic, but there are pathogenic microorganisms, and a lot of the fungi are going to fall into this category, but not necessarily. So again, we have to know the context. Did I skip one? Now we go up to the next trophic level and we get to the protozoa, and these are really fun to see in the soil. That's a flagellate right there. I'll show you some more videos. This is a naked amoeba right there. This is a testate amoeba. Testate just means it has a shell. These are live microorganisms, and you can see them in action under the microscope. you don't really have nutrient cycling going on until you get the protozoa in there. If you just have bacteria and fungi, you're not really going to have that nutrient cycling, because the protozoa are eating these guys. They basically are eating more nitrogen than they need. And Dr. Ingham likes to call it the poop loop. You know, these guys are eating the food, pooping it out. These guys are eating that. These guys are getting more. They're bigger, and they're pooping that out. And that's the nutrient, part of the nutrient cycling process. Among the protozoa, we do have ciliates, which do prefer a more low oxygen environment. So again, it's just another indicator that you're probably going anaerobic. It's not always a bad thing, though. You're going to get more ciliates if you've had like a big rain event. And then we get to the nematodes. A lot of people have heard of the root-feeding nematodes. They're very destructive. There's a lot of chemicals around trying to deal with root-feeding nematodes. So they are undesirable. But look, there are 4 other functional groups of nematodes that are good, that are extremely desirable. And here we have a bacterial feeder. This is a fungal feeder. We identify them by their mouth parts mostly. If y'all wanted to get into those details, we could, but we're just looking at the function, looking at their mouth parts and other body parts to determine, is this going to be functionally a bacterial feeder, a fungal feeder, predatory. Predatory are great because they're going to eat your root feeders. And I don't have videos personally, but there are videos on YouTube of them eating the root feeders, and they're just ripping them apart. Then you have the omnivores, which are what we call switchers. And so in this video, you can just kind of see that really big mouth part down at the bottom. They have a very wide mouth, and it's just very practical. They have a big mouth so they can stick big things in there. Let's see. Let's go on to. So now we're into some of the videos. And I'll just kind of talk through what we're looking at. This is a ciliate. He's very large. They're super fast. So when you're looking at them under the microscope, sometimes you'll just see a shadow zoom across, and you know that you probably got a ciliate in your sample. But this guy is going to come back up, I think. There he goes. They're really hard to follow. And these are soil aggregates. These are good signs. You can see the color is going to indicate to you whether they're humic or fulvic acids. The darker is going to be humic. The lighter is going to be fulvic.
So what does fulvic mean?
Let's see. Fulvic acids are just a less complex. So you've heard of humus, right? Humus is a good thing. We want humus in our soil. The humic and fulvic acids are foods. They help build that aggregation. A lot of times, some growers will use straight humic acids, and fulvic is just a less complex acid. So the honey colored is going to be basically, you're looking at a kind of a pathway, right? You're going to see fulvic acids first, and then as you get more cycling, more biological activity, you're going to see the darker ones, which are the humic acids, which are actually food for the fungi also. And then very dark ones, in some of the samples that I have, people are using biochar. I don't think that's biochar, but biochar will look very dark black like that.
Are either one of the acids indicative of the moisture conditions you talked about earlier, or like if it's oxygen rich or completely
Yeah, definitely.
deprived?
If you see, I think I might have an example of bacterial dominant soil here, but when you're looking at bacterial dominant soil, you're going to see no aggregation, none of those big chunks like that, and
Yeah.
yeah, it to me it looks dead.
Does it produces a compost that doesn't clump up into such a nice ball that it does if it has good fungal properties in it?
I mean
Is that
that
the case?
Yeah, that's really hard to give like a general guideline because um, there's so many different kinds of way to make compost that some have really clump a lot, like Johnson Sioux compost. I don't know if you are familiar with Johnson Sioux, but
Is it the bioreactor thing?
yeah, the Johnson
Yeah.
Sioux bioreactor. Uh,
Jack Spirko just did a video about it. I think he
yeah,
released it yesterday or the day before
right.
or something.
So in the Central Texas area, we're actually doing a lot with Johnson Sioux bioreactors because it is extremely biologically active. But compared to the soil food web methods, which is Dr. Elaine Ingham methods, which is a thermophilic process, hers are more consistent. That's just what we're seeing under the microscope. But the potential for the most biologically active compost is Johnson Sioux.
That's
Um,
interesting.
mhm.
Our Frenchman asked about earthworms in the soil. If he's seeing a bunch of earthworms, is that a sign of healthy soil, or
Oh, definitely.
is there a downside to seeing worms?
So it's definitely a sign of healthy soil because that means there's oxygen there, right? And they're actually improving that situation by tunneling through the soil, and they do their own kind of cycling. There is a downside. I honestly can't tell you the downside because it's only come out a little bit more since we've been getting more information on the soil food web. I, it's a rare downside, and I don't think most people have to deal with it. Um, so I mean, sorry, I don't know what it is, but usually I don't have a percentage, but very high percentage. If you have lots of earthworms, you have a good situation.
So we've just gone to an hour and 3 minutes. Are we okay with your time? We're okay with my time.
Yeah, I'm okay. Yeah,
All
I'm
right.
fine.
And then I've got some more broader questions to ask you here later on.
Okay. So this one is kind of a fun video. I'm just going to play it. Find the flagellate. You're just looking. Am I going to mess it up if I go full screen?
No.
He's a little guy right there in the middle.
You see him, right?
I can. Oh, okay. You're doing full screen. Okay.
Hopefully, it's going to lag, isn't it?
No, it's just a few seconds. It
Okay.
should
I'll
pop
be
up.
patient.
Yeah, this is this is a topic that I could talk about for hours. It's one of the most interesting things that I can think of when it comes to biology.
Finbear, how long have you been living on that piece of property that you're working on?
This is my third year.
Okay.
Yeah.
He just recently put a last year he put a greenhouse out back.
Yeah.
Yeah. It's occupied by my chickens at the moment because everything is frozen. So it's a perfect spot for them. It's nice and cozy for them because the greenhouse is it's like almost like a semicircle shape, like a hoop house.
Like,
So it's quite strong against snow loads, and it's covered like maybe half an inch to an inch of snow covering the entire structure. So the chickens keep it nice and warm, and I got like work lights keeping them lit during the daytime, so I get my
wow.
eggs. I get eggs and everything over the winter. But because they're a South American breed, they've slowed down a bit. So if I'm lucky, I get like 3 eggs a day at the moment.
Wow.
Yeah, I see the little critter. He's the fastest moving. He's the most traveled.
So this is really good. I'm pretty sure this is Johnson Sioux compost. So these are fungi. We're looking, a lot of times we look at the color. This one's like gorgeous. It's a nice dark brown color. It has septate. I don't know if you can barely see it. Yeah. They're basically the cross walls. I can't see them on my screen. You can see how this is lighter. When we're looking for fungi, we're looking for these squared off ends. If they're not squared off, usually it's going to be some kind of root material, and roots are also bigger. But yeah, you can see some movement here. That flagellate is probably under that aggregate. You can see these aggregates, more fungi here. This is just a beautiful sample. This one is. Let's go to the next one. I think, is that the same one? Yeah. Okay. So here we get to see a nematode in action. There's a flagellate going along right there. He's carrying some aggregate on his head. This is a nematode. Nematodes are notoriously hard to identify because they are really, really squiggly. But there goes that flagellate there. So it's funny, you can't really see his body because he has this aggregate that's on top of him and he's dragging it around. But just the way he's moving, we know he's a flagellate. They kind of bumble along. Sometimes you can see that flagellum.
So basically, that's like an XRP holder. Nobody knows what they're doing, but just sort of hang around.
Are these your videos, or
Yeah,
did you
these are
borrow these?
mine.
Okay,
No,
great.
no, these are mine. These are all mine. So let's see. Let's go on to the next one. Not very good. I love this one. This is from the Fungi Compost Company. And he does Johnson Sioux compost. You can see that flagellate right there, spinning, spinning, spinning. You could probably see, if you look closely, you can see a very faint tail going down into the drop. So imagine that we're looking at the slide, we're looking at the top surface, and that slide is going down 15 layers. And so you have these different depths. These are on the top layer, that's what's in focus, and this stuff that's out of focus is lower down in the drop. This is probably a microarthropod right here. It's a squished dead something right here. And these are all those beautiful aggregates that we're looking for. When we're looking at a sample, when we see something like this, it's like, oh, I might be at this for 2 hours because there's so much to look at. They're so fun to watch their movements.
How long do they survive on between the plates.
So the drop, it depends on your environment. Again, if you have low humidity, or if you have a breeze or whatever, it's going to dry out a lot faster. You can see it drying out because it starts kind of floating, and then the edges start kind of swallowing up everything. But
Is
but
this
usually
out in the landscape, or do you bring it back to the lab?
this
And
is in the lab.
what sort of petri dish, or is it literally between the slides, between glass?
It's between the slides.
Hmm.
So we go out, we take a soil sample, we bring it into the lab, we make a dilution, and we make different dilutions because we want, like, this dilution is probably a 1:8, because we don't want it too crowded. If it's too crowded, you can't really identify things, because just think on that slide, I guess the slides we're using are 15 micrometers. So, you're going 15 layers down. So we make a certain dilution, we put the drop on the slide.
Just with water?
Yep.
Okay.
Usually with water. You have to be careful about chlorine and chloramines and whatnot. And you can't use distilled water, because distilled water has something to do with the salt. Anyway, so I just use spring water. There also are slides. They're called well slides. So it has a little well in it. And so if you want to do a sample where you don't dilute it and you're trying to look for these micro arthropods, you can put it in that well slide and put the cover slide on top of it, and it's not going to squish it.
That's what I'm missing, I think.
Yeah. Okay. Yeah. So do you have a microscope, Davin?
Right over my shoulder.
Look here.
There it is, yeah. My kids got one as well.
Oh.
It's not
Yeah.
very high tech, but it's like you still see all sorts of orthopods and whatever.
Yeah, so we want at least to see 400x total magnification. So that means you would need to have probably a 40x objective, and then your eyepieces can be 10x or 20x. So if you have a 10x eyepiece and a 40x objective, then your total magnification will be 400. And that's most of these samples are at 400.
Yeah.
So I don't know. That was on, that we have a section on how to take a sample, how we take a sample. I have this in here because, back to, a lot of people are like, well, what can I do with this? I don't have equipment. How do I get the biology in my soil? An easy way is seed inoculation. And you can inoculate your seeds in a variety of different ways. Now, when I talk about seed inoculation, you have to be careful. We're not talking about, in the commercial seed industry, they're coating them with fungicides. You don't want that. Sometimes they're sterilizing them with bleach. No, because there's a microbiome on the seed. And then you can add more biology by doing a fermentation, by doing compost extract washes or seed soaks. If you want to really look into something interesting, Google Iroquois corn medicine, and they were doing this. Basically they would sometimes even spit on the seed, but they treated the seeds in different ways to get that microbiome into the soil. And what happens is the seed uses that microbiome initially to help it germinate, but then when it's done, that goes into the soil. So seed inoculation is extremely beneficial and extremely easy. If you are interested, we can talk about taking soil samples. And then, of course, this goes into what we do through this, certified through the Soil Food Web School, is we test the sample, we actually count the microorganisms. Of course, we have software that will extrapolate out that information and give you a fungal to bacterial biomass ratio. And then we can see these other things that we might add into the report. If we see, root feeders, if we see anything that's indicating anaerobic conditions, if we see all the levels, all the role players, then we know that you probably have nutrient cycling going on, unless you have some kind of very unusual circumstance. We can see the diversity of your organisms. We're looking at the diversity of bacteria, the diversity of the protozoa. So we saw a few flagellate shapes. They come in a variety of shapes, and the amoeba come in a variety of shapes, and we're looking for that diversity. And we can kind of see where you're at in succession and give you a comparison. If you say, oh, I'm just growing native plants, great. We can see you're probably good. And you say, oh, I'm growing fruit crops, trees. You've got a bacterial dominant, you have a very low fungal to bacterial biomass, then we know, well, you got a lot of work to do. And then standard deviation, that's just kind of how we keep our information accurate. If there's over 70% deviation, we're going to probably throw it out as unreliable information, because there's just too much deviation.
Can you give us a general understanding of where it is that you take the samples and how you take the samples?
How I take the samples? Yeah, taking the samples is actually pretty easy. If you've ever done traditional chemical soil testing, it's easier than that, because we're only doing the top 3 inches of soil. And so usually we just use an apple core. And let me see if the other one has the materials that we use.
But where do you select? Take it over there at the high point and then over here at the tree line?
Exactly.
Okay.
So it depends on what you're looking for. What information are you hoping to get? Let's say you're worried about this tree here. It's suffering. So you want to check the biology in that root zone. And so when we take a sample for a specific plant like this, or a large shrub that's sick, then you're going to go halfway between the drip line, which is, the outer canopy of the tree. And we're going to take at least 3 samples. We're going to put that apple core into the soil. You kind of, rotate it back and forth, get a good 3-inch core, put it in a little baggie, and take 2 more cores and mix that all in the bag. If you're going to do, that's labeling. Let me see. If you're going to, I don't know if it's in here, but let's say you just want to do your garden beds. So someone has raised beds. You're going to take 3 core samples from that raised bed and mix it together. If you have, you're going to try to avoid getting too close to the edges, just because sometimes there's more moisture in the edges. It's just kind of a unique zone. You're going to avoid those areas. If we're on a larger property, let's say we're doing, usually it depends on the size of the property. A 1 acre property, you're just going to do like a grid, and you're going to take more samples. If you're, and you're going to avoid unusual areas. If you have a divot, if you have a puddling area, you're going to avoid those areas. You're going to basically, it has to be a random sample. You can't go and say, oh, everything looks really good here and really good here. No, that's not going to give you a representative sample. It doesn't need to be random, but you should avoid unusual areas. Maybe you have a truck that goes through the property and it's constantly making ruts. You're not going to take the sample from where the ruts are. But if you have a situation in which you're struggling and you want to find out what's going on in this spot, then yeah, you would take your soil sample from that, a huge ponding area or whatever. Yeah.
So, can you talk a little bit about your background of why it is that you arrived where you are and why people give you a call to take these studies?
Yeah. So I'm a certified master gardener. I'm in my 10th year. Once I became a master gardener, I've realized that I knew more than already. And so I began to do consultations, and eventually I started doing landscape design. I focused on what I would call ecological landscape design. And then, and I always gardened organically, and I would have people that wanted an organic landscape, I would have to tell them we're looking at 3 to 4 years to get the ecosystem functioning properly. And it just turns out a lot of people don't want to wait that long, and they can't wrap their mind around an ecosystem. It's a holistic system. And so they just think organic is just changing your inputs, right? Oh, I used chemical fertilizers before, now I'm going to use organic fertilizers. And no, that's not what it's about. It's about having a functioning ecosystem. So I found out about the Soil Food Web School, and I just thought, wow, I mean, this is how I get the information in front of people, instead of trying to mentally walk them through. And I tried all kinds of different ways. We had so many different projects, and, they would just kind of fizzle because it was just so hard to get someone to fully transition. And
because you're not giving them a foundation of learning. Instead, you're just like plopping the information that they don't understand the beginning or the end.
I think
so they don't respect it, right?
Yeah, exactly. I think a big thing when you see these microorganisms, all of a sudden you realize we are talking about life. And we're not talking about how to get rid of that weed. We're understanding that weed is an indicator species. It's telling us something about our environment. It's telling us something about our management decisions. And we need to work with what it's telling us. We don't need to go find the best organic solution to get rid of that weed and kill it. No, we need to find a way to cultivate the life in the environment. And I think I don't know a lot about permaculture. I just know some stuff, but I think there's a concept that y'all talk about. It's when you talk about energy in the system, right? And there's a finite we have a carrying load, right? We have a finite amount of carbon in the system. We have a finite amount of phosphorus in the system, and we have all these cycles going on in our environment, and we're not respecting those cycles, and we're draining energy from the system instead of cultivating life, which is what regenerative ag is, right? I don't really like to say I do regenerative ag, but the principles are the same. I just really like to stress that we're doing a biological approach that fits with organic, it fits with regenerative, it fits with permaculture, it fits with biodynamic. It's all about cultivating the life in the system. And I think that you can really awaken people's mind to that when you show them the life, right? When you look at those microorganisms and you start seeing their personalities and their characteristics, right? It's the same with the nutrients. I love soil experts that talk about the behaviors of nutrients in the system. They behave in certain ways. They have personalities. And I think when people start seeing that, it starts dawning on them, oh, this is the life. This is what we're doing. This is why we're protecting our soil.
That's
You know.
lovely because it's out of sight, out of mind. They don't see it
Right.
microscopic.
Right.
That's wonderful.
Yeah.
And then so people come to you when they have a certain need, and then we could also relate this to how you're helping your brother.
Yeah. So my experience, I thought, oh, I've done landscape design and consultant for all these years. It's just going to be an easy transition. But what I found out now that I'm certified and I'm getting this stuff in front of people, I just can't tell you. I have an industrial composter and taking biosludge from the city and, 97 acres, and they're trying to make good compost with toxic waste. I have a Johnson Sioux composter. I have 2 ranches, farms, I guess I say. One is my brother that you mentioned. Another one is, they're trying to transition completely from probably a century of conventional corn growing on their property to regenerative. And right now they're at the stage of they did nothing. And we're looking at the biology. And that's so interesting. My brother kind of did the same thing on his land because he had improved pastures. And he's like, what do I put on? Nothing. And we've been looking at that biology, and you can see how it's healing. So we're really looking at a lot of off-the-shelf products now. I'm in another microscopy group where we're really, because that's the public is getting inundated with those products, and nobody knows really what they're doing. So we're starting to do studies and what is the gardener in our area, what are they using, and looking at those off-the-shelf products and comparing them. And what else? It's been kind of crazy. Oh, and the main thing that I'm probably doing is giving workshops, because there's so many people like you, Davin, who are getting their microscopes. They have the setup, but they need more coaching on to develop that visual literacy of what they're looking at and what they're seeing in their samples. So I'm doing more of those kinds of things. Yeah.
Awesome.
Yeah, this has been like really interesting thus far. I was wondering, did you guys talk about us doing this again at some point? Perhaps, hopefully.
No,
What do
we're
you
trying
say?
to get every question we've got right now. We're trying to do it. So I want to know how many different scientists are there floating around these same ideas?
I couldn't tell you how many. I can definitely tell you a lot of the big players. And I have a YouTube playlist that has a lot of these important videos in it. You're talking about Dr. James White. So all you have to do is Google his name and look at YouTube videos, and you'll see how much he's putting out there. I actually just attended a live session with him right before this because he was speaking in Nairobi, I think. Not in Nairobi, but to a group in Nairobi. So he's someone to look at. The book that I showed you, he's not a doctor. He's actually a journalist, Jeff Lowenfels. His books, so that Teaming with Bacteria book, there's actually a whole series. And the first one was Teaming with Microbes, and it came out in 2006. all you have to do is open that book and you'll see a lot of references. He's mostly chronicling in that book Dr. Ingham's work that she was doing at that time and pushing it out to the public. And then he wrote Teeming with Nutrients, Teeming with Fungi, and then this last year he came out with Teeming with Bacteria, because all that new information about the rhizophagy cycle came out through Dr. James White. He didn't discover it, but he's the one who really took hold of it. And he's from Rutgers University and has really been pushing out a lot of research on that. Then there's Dr. Christine Jones. There's Nicole Masters. There's Ray Archuleta. There's, of course, Gabe Brown. Yeah, those are a lot of the main players.
Is it late Gabe Brown now, or am I mixing up the fellas? I think he
Father and son who are doing cattle, right? And
Gabe Brown.
no, okay. I think I have it mixed up with.
Yeah, I think I've seen some recent videos that he's releasing. I would be surprised that I hadn't heard.
Yeah. I think it was somebody.
G-A-B.
I have him.
Gabe.
Yeah.
Mm-hmm.
Yeah, I know. I've seen him with Ray on some videos, I think.
So, what do you think that are some things that our society has to get through in order to go to like a timeline that respects the health of the soil rather than pumping it full of chemicals, getting what we can out of it, abusing the soil?
Say it again. You're asking about how long that would take?
No, I'm saying more like what sort of aspects do you think that our society has to respect from the land to respect the timeline of life cycles? Because right now we're pumping chemicals in, or we're putting those raw materials into the land in order to harvest as much as possible.
Mm-hmm.
And it's not allowing natural life to take place. So we're abusing the land,
Mm-hmm.
all based on our expectations. So like what sort of step back of expectations with society do we need to look at?
Yeah, so I have my hands in a lot of different projects along those lines. So I do the soil food web assessments, but there's also carbon in the US. It's called carbon farm planning. Basically what we're talking about is applicable to the landscape industry, to commercial and residential landscapes. It's applicable to natural areas where they have, in the US, we have NRCS, which has conservation practice standards, and there's a lot of grants available. And it's just about doing things like hedgerows, pollinator areas, riparian restoration, compost applications. I think there's like 35 plus different practices. And actually I have to say, if I was speaking to my tribe, the people that I really get into different things about, we're going to talk about this on an even higher level of carbon sequestration. And how can we get that misplaced carbon? We have misplaced moisture. The whole climate conversation is kind of messed up right now because they talk about emissions only. They don't talk about how we've messed up the water cycle. And all of this is directly related to that, because the benefits of having a healthy soil food web is you're gonna have what is called the soil sponge. And you can look up D.D. Pursehouse and Walter Yena, and they talk about the soil sponge. And it's basically the idea that you're getting that aggregation. You just think about a sponge. It's keeping that moisture in the ground, and it affects the water cycle. And there have been studies in the US, there have been large ranches that get more rainfall than surrounding ranches because they've been practicing these soil health principles, and that's documented. So they're affecting the water cycle. So I mean, to me, I mean, I can get pretty frantic when I start talking about that, because people call it climate change. I call it a climate emergency. It's like, wake up, people. But this is how we're going to fix it. By recovering our degraded soils. This is a huge player in the game, and there's all kinds of, this is beyond my knowledge, because these are climatologists and that have run these numbers, and that the research is there. We know how to fix this predicament we've gotten ourselves into, but we need more people on board. So I just talked with someone who's going to get me in touch with a billionaire who bought hundreds, I think, one of the largest land sales in the U.S., and they're in Montana, and trying to get in touch with them, and they have a charity organization, and let's see what we can do. What are they going to find there on their land? So I don't know. Does that answer your question, Davin? Is that where you were going with that?
I actually forgot what my question was, because I'm gearing up for the next question.
Okay.
But I also come from the camp that thinks that the scientists are paid to come up with the theories that they have. And so these politicians that go around like Al Gore and say that things are going to change, and that like the waters are going to rise there in New York City, I just want them to tell me exactly when they're going to, come into the city, because it hasn't changed a bit in the last 100 years, 200 years, and we have markings on rocks that are there on the coast. So like, these people in Martha Vineyards, they're buying these islands, but yet they're telling everybody that those islands won't exist anymore. However, nothing's changed. And so I think that they're just playing political games with us, and they're manipulating stuff that interacts with us, like the weather. I believe that the weather is completely modified. So I just needed to push back just a little bit on you saying that the scientists, there's tons of data. I think that the scientists wouldn't get paid if they didn't side on that,
Yeah, fair. That's fair. And I will tell you, the scientists that I'm talking about, they're not getting paid. I'm talking about the scientists that have been, what do you call it? Ousted.
Oh, okay, cool.
Yeah. And and I can get those resources together for you. I'm not, and as far as, the rising sea levels, I can't really comment on that. That's not the stuff that I'm talking about. I'm talking about more the weather patterns. Okay. And the droughts, the extreme weather patterns, which have been documented. It's just, they're becoming more extreme. And when you listen to someone like Walter Yena, it's Jehne, he explains that very well. I think I'll send that stuff your way so y'all can look at it.
And we have this recorded, so I'm going to go back through here and compile some notes. I wonder if it is, I always like to look at scales of things, 10 times 10 times 10 in both directions. I wonder, by looking at the soil, have you learned, or do you think that people can learn more about themselves by looking, at the food web?
Yeah, I love that question. I think yes. I mean, I've always done that as a gardener, just to understand human nature more by uh, observing plants and the behavior of plants in the environment. And I believe it's true with the microbiome also. Um, there's a lot of I'm not fully into this realm yet, but there's a lot of people that are talking about the comparing the human microbiome to the soil microbiome, and talking about soil food as medicine, and even some places that are, I don't know what the names of what they're called, but basically they're like a community center where people are actually able to kind of have like prescriptions, and their prescription is to eat nutrient dense foods. And so yeah, I think there's a lot to learn about ourselves by looking at the soil. What have I learned? I don't know. I'd have to think about that a little bit more.
It's pretty much just engulfed you.
Yeah.
You've become a beast in the jungle sort of thing. So I had another question
Yeah.
that was like, do you think that we need to preserve natives, or should we allow to, because you were talking about changing the soil, and how, like, if it is that you're planting natives in a certain spot, then why improve the soil, because the natives won't adore that soil anymore. So I wonder if, like, if we're thinking too short span of a timeline whenever it is that we're respecting natives, and instead we should like, there's always going to be these placements where they're happy, the zone where they're happy is going to move. So if we put a lot of work into making the soil more and more fertile, whatever we want, well, still on the edges that we don't get around to, there's still going to be a perfect place for those natives to thrive.
Yeah, I don't know if this is where you're going, but when I think about that, I think so as a,
Because, like, if we look at a certain area that's just like New Mexico, or the desert, or just anything like that, um and then people say, well, keep it the same, and it's like, well, why wouldn't you allow it to change a little bit?
right. So I mean, just first off, I want to say, as far as natives versus adapted plants, and of course, food crops are not natives. There are people all on a spectrum of how militant do we have to be about preserving the natives. And I'm not at either extreme, because plants do adapt. But the thing about natives that you have to remember, I'm going to give you an example by going back to the rhizophagy cycle and these microorganisms. These relationships that they form with plants are very unique. So you have certain bacteria that prefer certain plants, certain mycorrhizal fungi that associate with certain plants. And these are associations that have been developed over, thousands of years, right? And so the adaptations do happen. But there was a guy out of A&M that I listened to. They were doing research. It was the first time I actually heard someone acknowledge that the genetics of a plant matter. And because of the weather events that we've had in Texas the last 3 years, A&M is scrambling because they don't have any historical data to compare it to. And what they're seeing, though, is that the climate change is happening too fast for these species populations to adapt. So they're not adapting fast enough. So, just look at the oaks in Central Texas. So many of them have been damaged by these last freezes. And historically, we don't have the data. Did it happen, way back when, before we were around? Probably, but we don't have the data, and the plants are not able to evolve fast enough. The normal natural selection, the adaptations that they do doesn't happen on the time frame that it's gotten compressed to. And that's why some, ecologically oriented people are, panicking, because we see this, the wildlife biologists, the plant people, we see that these plants are not adapting quickly enough. And so, so that, to me, that's the issue. It's going to happen over time, but our system is so messed up right now that, it's too dramatic of a change. Does that make sense?
Yes. However, what I was wanting to attack was this idea that we should keep things the same,
right?
Instead of promoting life and, understanding that there will always be space for those types of plants.
But I think that that's the point, is that there won't always be a space for those kinds of plants, because when we're talking about the plant, we're not just talking about the plant, we're talking about the microbiology, we're talking about the pollinators, we're talking about a very complex system that humans like to come in and make these little tweaks and not realize that that tweak has this, exponential effect, because now all of a sudden you've had an effect on the pollinator species, you've had an effect on the predators. And so that's when you have these situations where you have a competitor come in and it takes advantage of the situation. And that's what non-natives do. That's the problem with some non-native plants, is that they're too aggressive. So just think of the common example is kudzu. It's overtaken the South in Texas.
Which is
it's a
the vine.
monoculture. Yeah.
It's the vine that reaches to the tops of trees and suffocates them from the sun.
Right. And it's a monoculture. You can see acres of just, these shapes covered in these vines and everything dying. And a monoculture is not conducive to a functioning ecosystem.
Mm-hmm.
We have to have a polyculture. We have to have diversity. That's how nature works. I think that something to mention here too is that with the soil food web, basically we're talking about natural laws, right? That we have an ecosystem that functions according to laws, and those are called natural laws. And when we apply the inorganic fertilizers, when we do tillage, we are not abiding by those laws. And so that's how we're corrupting the system, because we're not abiding by the natural laws of that system, how they normally function. So when you look at the rhizophagy cycle, you see that, oh, look, everything is beautiful. It functions so well. But when we put some kind of disturbance on the system, or we put inorganic fertilizers, now we've disrupted that entire functioning ecosystem. Because what happens with rhizophagy, if you apply inorganic fertilizers, you basically send a signal to the plant to stop doing what they're doing. It's like males who have low testosterone, they don't want to give a young male testosterone because he's going to stop making his own testosterone. So they're going to give him something like Clomid. Fine, give the old guy testosterone. He doesn't need that system to be. He doesn't want to reproduce probably, but the body stops doing what it's supposed to do when you put something to take over that normal functioning. So that's what we're doing in the environment when we don't, take care of the native ecosystems that are already there.
So I don't know if Tin Top over here in the chat is being serious or not. I don't know if this is a joke, but he says, so is Texas at risk of becoming American Sahara?
Yeah, definitely. Yeah, there's a lot of people worried about that. I mean, it's happening over and over again already. I'll be going next week. I'm going to Lubbock, which is North Texas, and it's very dry there. And basically, it's John Kemp. Oh, he's someone else in this space that y'all have to listen to if you're not familiar with John Kemp. But it's basically how to farm in a dryland environment, because we're as much risk as not having any water as California is. California, Nevada, they're already experiencing this. And yes, there are certain parts of Texas that are very susceptible to just the Dust Bowl all over again. Yeah.
Well, do you have anything else that you'd like to say to wrap up this whole interview? And thank you so much for being here. We are going to spread this all across our crypto community.
Yeah, I don't think I do. I mean, we covered a lot of stuff. I just want to say thank you to everyone. I mean, your questions have been really good. It's so fun to talk about this stuff with people who are interested and engaged. I could talk about it all day. Actually, I do talk about it all day and all weekend and all night. So yeah.
Finn Bear had to go put his kids to bed, but he says, thank you, Laurie.
His kids would love this. Kids are great to teach this to. Jeff I'll leave with this. Jeff Lowenfels, if you look up his videos on the New Soil Food Web, he always makes this comment that all of our kids, at least in the US, they're so crazy about dinosaurs. They know these crazy dinosaur names. They memorize names that I can't even pronounce or spell. They don't know the names of these microorganisms, of the bacteria, Pseudomonas. Why are we not teaching the kids all this stuff? I mean, they love it. They see this and they're just like, and then they start saying, why are the adults so stupid?
Thank you, Laurie.
Yeah. All right.